Hi there, this video outlines some variations on viral structure, including the helical, icosahedral type shapes, as well as envelopes and some optional structures. So the helical virus is somewhat cylindrical when you look at it in a very sort of simplistic view. And remember that the capsid on the outside is made up of capsomeres. And then inside of that is the nucleic acid. So remember, that could be single-stranded DNA, single-stranded RNA, double-stranded DNA, double-stranded RNA. So I'll just kind of remind you that that capsid is a coating. So that's in the interior, just kind of having that extend out a little bit there. But then you also can have an enveloped type. So an envelope type tends to be, that's helical, tends to be a bit, quite a bit longer, very flexible. And so if we draw the envelope in, and remember that envelope is a phospholipid bilayer, it's actually picked up from the host cell. So these types are considered to be animal viruses. And on the outside, you'll have an envelope, and then I want to identify those same structures again. You very often have spikes as well on the outside. And spikes appear as these little projections. Those are extremely important for binding and attachment to the host cell. All viruses would have some sort of three-dimensional structure for binding. They just may or may not have these specific spike projections. Okay, so again, going back to that helical shape that's there, so now you're going to have this kind of rolled up sort of tube that's on the inside. So I'm using blue again for the capsid. So again, here's our capsid, that protein coating, and then inside of that, of course, would be your nucleic acid. And of course, that's on the interior, so I'm just going to remind you of that by covering that. And remember that capsids are made of capsomeres. Those are the protein subunits. This tends to be a softer, more flexible. The envelope is picked up from the host cell membrane as it leaves the membrane. It actually pinches off and comes with it. So this is actually host cell membrane that's coming with it. The spikes themselves, the genetic sequence to make those would be contained within that nucleic acid. So those proteins would be synthesized during transcription translation. And then those would go to the host cell membrane, kind of get embedded, and as the this capsid middle here passes through the host cell membrane, it would pick up the envelope, and those spikes would be there, and that would come with it as well. So that's the helical type. So now let's compare that to the icosahedral type. So it's a 20-sided, I'm not going to effectively show all 20 sides here, but 20-sided figure. And again, it's still made of capsomeres. So here's our capsid. And again, on the interior is going to be our nucleic acid. Could be either double or single-stranded DNA or double or single-stranded RNA. Okay. Okay, so let's compare that very quickly to the enveloped version. So kind of same thing. We've got our 20-sided structure. We've got our nucleic acid inside. And then there's our capsid on the outside. Now we're just going to add that envelope. So that's really about the same. So I'm going to draw this, kind of making it look a little bit flexible because it's this soft phospholipid bilayer. And would have our spikes, possibly. And spikes are an option, but there's going to be some sort of bumpy, again, three-dimensional surface. Keep in mind that spikes can also come right off of what's called this naked naked virus as well. So this is our naked. Up here, these two, meaning they lack an envelope, and here's our enveloped versions down here. Okay, so again, I'll just quickly label the spikes. Those are those projections for attachments, and again, the envelope. And the same thing, so the release is going to be the same. So as this virus again leaves, the capsule will pass through, pinch off the envelope with the spikes that are projecting. So quickly to recap, when we talk about viruses, one of the ways we can classify them, one of the big ways, is their structure. So we can also look at their genetics and the diseases that they cause. But the structures can be naked or what we call enveloped. And then of the two naked types, we can have the icosahedral type or the helical type. And then you can have enveloped versions, and that's where it has this additional protective coating. So again, this is just the helical type down here. The naked helical tends to be a bit more rigid. It can be a little more soft and flexible when it's got that protection of the envelope that's there. Now there are some other complex types of viruses that I'll cover in a different video. It doesn't mean that they've totally thrown out the rule book here, but they just have some additional structures that make them a little bit different. Okay, that's all I have on those. Please contact me with any questions.